biodiversity life with a major breakthrough in the island of Texel in the water sea. Let's quickly move to our call.
Well, thank you, Zora. So as you can see, we're here on Texel. I'm here with Dr. Lars Bootsma, and he and his team have solved one of the most fundamental scientific mysteries of the modern age. They spent 5 long days to determine vegetation composition along environmental gradients. It's incredible what you have achieved in the last 5 days. How do you feel? As you can see, he's very happy.
Well, thank you, Lars. Well, plants tolerate a narrow range of salinity. Due to human drivers, climate change, floods, and other aspects have been rising. These are major influences on small islands like Texel. By filling the knowledge gap, we can find out which plants tolerate what kind of environmental conditions to help predict where they might grow in the future. But Binnen, what does plant zonation actually look like?
Right, so to make clear how important these salinity gradients can get, this little stream is a passage for salt water. And you can clearly see that one type of vegetation dominates this little piece, whereas here there's more lamb's ear and short grass. When you go further away from the salty water, there will be higher grasses and other types of vegetation as well, which is
That's so cool how you can see the different types of zonation. But my question is, how do you measure the different types?
In the slaughter, 18 measurements were taken. These were divided over 3 transect lines. These transect lines were placed along a creek. This creek came from the sea and went more land inwards.
First of all, we need plants. Here, there are plants. We're using these kinds of cards to see which plant is which. Here we have creeping willow. There's also a lot of creeping willow here. For instance, in this little block right here, there's about 30 to 40% coverage of creeping willow.
By measuring these tiny little squares in this big square, you add some of it and you end up with the composition. That's so interesting. But then I wonder, how do you actually measure the salt? Do you lick the ground, or I don't know? So that's why we go to our soil expert.
So, welcome to our soil lab. Here we have all the different soil samples that were collected, and then, as you can see, we weigh out 15 grams in these secret containers that are all labeled, and then we add 75 milliliters of water, shake these up really good, then we measure the salinity with this EC monitor.
Now for our data analysis. Let me take you through our thought process. We are dealing with numerical input variables. We want to know the correlation between salinity and vegetation composition. This is why we chose a spearmint test. Our data includes a lot of zeros. This is because we measured some species in a few transect points while there were none of those species in other transect points. The spearmint test takes these zeros into account as outliers. This is why these zeros will not mess up the results. We performed this test on all species. However, the results showed that there was a weak correlation. This does not support our hypothesis. This is why we conclude that we definitely saw zonation, but to actually conclude it, more research should be done. Let me take you through our results. As you can see, in transect line A, the salinities were the lowest. This agrees with our hypothesis because there were plant species there that were not present in line B and C, where the salinity was significantly higher.
Oh, so in that graph, you see all the vegetation against all the salinity levels. But then, how do you see this per one species?
That's a great question. As you see here, on the y-axis we have C wormwood, which is a species tolerant to salinity, and on the x-axis we have the salinity rate. As you can see, the coverage grows a little bit with the salinity. The researchers have actually also did something extra, which is measure the biodiversity. As you can see, on the y-axis there's biodiversity, and on the x-axis is soil salinity again. And you can see that the biodiversity actually declined with the soil salinity.
So you basically got no conclusion. Well, that sucks. Let's see how Lars feels.
We couldn't take enough measurements, We had no time, we couldn't get close to the sea, and we got no correlation at all. Yeah, that sucks.
For follow-up research, we recommend using more transect lines, more measurement points along the transect lines and along more creeks. This would get more results for more valid data.
Well, you heard it here first, folks. Till next time.